Hello and welcome dear viewers to the informative topic on force degradation studies in pharmaceutical industry, and in this video we are going to understand the force degradation studies in very much detail, and after watching this video you will be able to understand all the aspects of the force degradation studies, like why the force degradation studies are done, how these are done, and what are the requirements as per regulatory authorities, and what are the applications in the pharmaceutical product development along with the analytical method development. So force degradation studies, these are also known as FD studies sometimes, or stress testing studies, or these are also known as degradation profiling studies, and also stress decomposition studies. So the force degradation is a study which involves the testing of effect of different stress conditions applied to the drug substance and the drug product. So one point here to understand is that these force degradation studies are done on both the drug substances and also on the drug products. The FD studies involves degradation of the drug product and drug substances at the conditions which are more severe or more stressful than the accelerated conditions, and thus these studies lead to the generation of the degradation into the drug substance and the formulation. So these studies are done to determine the stability of the molecule. Generally, the stability studies involve the storage conditions like long-term condition, intermediate condition, and the accelerated condition, which are 25 degrees Celsius, then 30 degrees Celsius, and 40 degrees Celsius at the humidity level of 75% RH. But here in the force degradation studies, the temperature above 40 degrees Celsius can be used, and the drug substance and the products are exposed to that temperature so that the thermal effect can be studied onto the intrinsic stability of the drug substance and the formulation. So these stress testing is also known as stress degradation, stress testing, stress studies, stress decomposition studies, and the force decomposition studies. Also these are called as FDS and degradation profile study. The force degradation studies uses the stress conditions to create the degradants in the drug product and the drug substance. So here the stress testing involves the conditions which are more severe than the normal and formal stability studies conditions. So that we have to understand in detail. Also, some of the conditions are very different from the formal stability studies, like uh, stress by acid and base, stress by peroxides and metal ions. Then why these FD studies are required? So the stability of the drug substance and drug product under accelerated condition or at a condition more stressful than the accelerated condition is required to be determined. The results of the FD study helps to develop the formulation composition, the manufacturing process, and also helps to develop the packaging system for the drug substance and the formula. Container closure and pack configurations are also can be developed based on the outcomes of the force degradation studies. These studies are also sometimes used to define the storage condition of API, intermediate stage materials like bulk materials, lubricated blends, and also the finished product. And these studies also help into the shelf life. These studies are done to solve the stability related problems also. Then why these FD study is done? So this study is done to study the chemical stability of the formulation and drug substance. That is the intrinsic stability. Intrinsic stability is the nature of the molecule, whether it is a unstable molecule or whether it is a rock stable molecule, that can be understood from the force degradation studies. Then determination of the likely degradants, because in the stress testing the severe conditions are given to the drug substance and the formulation, and that's why it can give a complete information about the likely degradants. Then these are used to detect the degradation impurity generation pathways and their mechanisms and the conditions in which the degradation happens. Then it is done to study the degradants impurity structure, to develop the stability indicating analytical methods, to establish a degradation profile as a regulatory and ICH requirements, and to comply that requirements, these studies are done. To develop a stability indicating methods are specifically designed to separate drug degradants from non-degraded drug. That means the generally known uh, terminology is there, stability indicating method or stability indicating assay method. So these methods are developed and validated and tested by doing the FD studies. Then these methods are required to be stability indicating, and that justification can be given on the basis of the outcome of the FD studies. So when this study is performed, generally FD study is for the development of new drug substances and new drug products, and also the generic formulations are required to be performed for the force degradation studies. FDA recommended that it can be performed in the phase 3 of the regulatory submission process, and in phase 3 it is a clinical phase where the regulatory proof is provided for the drug molecule or drug candidates for its benefits in a large targeted patient populations with the specific disease or specific indication. The impurity degradation basics are there. Some factors are there which have a great impact on the generation of the impurities, and these are exposure to humidity or moisture, exposure to pH, exposure to temperature, that may be the storage temperature also, then exposure to the light, and combination of the any of these above listed conditions like temperature and RH, temperature and light, light and humidity, pH and temperature. So any combination can also have a impact on the drug substance and the drug product for its degradation. Then degradation routes involve the major conditions like hydrolysis, which may be due to acid and base, oxidation, oxidative degradation, heat. Heat may be dry and moist heat, that is also called as thermal exposure or thermal degradation. Then there is a exposure to the light, that is called as photolysis or photolytic degradation. And a new condition for the forced degradation is given by the ANVISA from Brazil. It involves the use of metal ions to test the susceptibility of the formulation of drug substance to the metal ions. The stress test with metal ions such as Fe and Cu should be used to assess the drug substance susceptibility to metal catalyzed oxidation. As these metal ions catalyze the oxidation process, that's why these two metal ions susceptibility is required to be studied, and that's why due to this oxidation degradation may happen. So ANVISA gives the information about the metal ion catalyzed oxidation. Then the stress conditions are there like hydrolysis, which may be by acid or base, and in experiment of the hydrolysis the acid is used and the base is used and the pH is changed so that the effect of the acid and base can be studied. So this acid base or pH impact can be studied at room temperature, and if at room temperature there is a very less degradation or degradation is not happening, then the temperature can be increased from 25 to 40 or 60 or 80 based on the molecule property, and sampling can be done from some hours to some days. So these conditions are defined based on the property of the formulation and the property of the molecule, that is the sensitivity of the molecule to these conditions. Then oxidation, it is generally done with hydrogen peroxide, and AIBN is also used. Other oxidative materials can be used and if justified. they can be used for the oxidation reasons. Storage conditions are 25 degree, 40 degree and 60 degree as per the requirement, and the sampling may be done from some hours to some days. Then photolytic. So photolytic condition is given in the ICH, and it involves the 1.2 million lux hours and 200 watt hours per meter square light exposure as per the ICH Q1B. So this is the normal requirement for stress testing. So these exposure can be done two or more times, like 3 times, to study the stress effect of light. So after the required illumination time, the sample is removed and then the impurity analysis is done. So in all these conditions, the as such API is kept and placebos are also kept. So then coming to the thermal degradation. Thermal degradation involves dry heat and moist heat. So temperature may be from 40 to 80 degrees Celsius, and also the sampling may be performed in some hours to some days. Then coming to the metal ions, generally ferric oxide and the Cu with two positive ions can be used for these metal ion studies. This may be done with the transition metal ions, and the effect of the transition metal ions on the degradation profile can be studied. Then at last humidity can be used. So the humidity chambers are also available, in which the drug and the formulation is kept and the effect of the humidity is determined. So from low to high humidity conditions are given to the drug substance and the formulation, and its effect is studied on its degradation profile and also on its physical description. Then these guidelines are there for force degradation. Mainly the ICH guidelines are there, FDA guidelines are there, EMA guidelines are there. USP and Japanese pharmacopoeia also gives the information about the force degradation. And currently, ANVISA has given a new guideline which involves the metal ion catalyzed oxidation study. Then what are the limits for the force degradation studies and what is the acceptance criteria? So generally there is no such acceptance criteria for the force degradation that this much degradation should be there. But in general, 5 to 20% of the degradation is accepted, and it is required for the validation of the chromatographic assays, so that the applicant can prove that the analytical method is stability indicating, and all the impurities are studied, and the understanding about the molecule stability and sensitivity is studied. The stability should be more than 90% and about, hence 10% degradation is considered enough. See, the assay limit is 90 to 110%, and if there is a degradation of 10%, then it will give 90% of the assay. So active degradation target and study determination at the FDS conditions. It is specific ANVISA requirement that each FDS condition must produce a degradation greater than 10%, but lower than that one, which would lead to a complete degradation of the sample, compromising the test. So more than 10% as per ANVISA, and generally 5 to 10% or 10 to 15% or 20% is generally accepted by the regulatory authorities. So here is the example. It is a hypothetical example you can consider for drug degradation for drug substance or product. So for hydrolysis, I have shown a degradation of total impurity 6.55%, and assay is coming 93.23%. Then oxidation, I have considered total impurities of 14%, and this impurity generation is due to increase in the impurity B. Then I have considered thermal degradation with 1% for impurity A. So this is a hypothetical example. Like this, the degradation study results are shown into the table by the applicant, and mass balance is given. So mass balance is nothing but how much is the total degradation and what is the assay. That mass should be get reconciled, that is known as the mass balance. Like here for 6.55% total impurities, 93.23% assay is coming, and for 14% of the total impurities assay is coming 86%. So that you can say that the analytical method is stability indicating and degradation is achieved more than the 10%. Then coming to the mass balance, loss in assay is equal to total impurities, degradation, that is degradants. So that loss in assay is total impurities. Mass balance is required as per the part of the study, and 95 to 100% mass balance is acceptable. If mass balance is not achieved, then justification is required for not establishing the mass balance. And generally, it is not a thumb rule that you will achieve the mass balance. Sometimes because of the different impurities or degradants, their behaviors and their analysis, sometimes volatile impurity get generated. That times it is difficult to have a mass balance. Mass balance may come on lower side or maybe a little bit on higher side. So mass balance determines the formation of the impurities or degradation product, and it is required for ensuring the safety and efficacy. Also, if there is a sufficient mass balance is achieved, then we can justify that the degradation is achieved and the applicant may give information or confidence uh, regarding the study to the regulatory authorities. Then coming to the applications in the pharmaceutical R&D, and these applications are mainly for the formulation development, and also these principles can be utilized for the drug substance manufacturer. So if degradation is due to hydrolysis, then for the formulation, select the excipient based on the pH, whether it is acidic or basic. Use acidic or basic ingredients to stabilize the drug substance in the drug product. You may use acid like citric acid, or you may use base like sodium hydroxide or meglumine. Protect from moisture, keep at cool and dry place. Use dry granulation process, do not use water in manufacturing process as water may due to hydrolysis, and use solvent based granulation and film coatings so that water can be avoided. Then we have to select the packaging materials based on the oxygen transmission rate, that is OTR, and vapor transmission rate, that is WVTR. So these based on this OTR and MVTR testing and specifications, the packaging materials can be finalized. Then if degradation is by oxidation, then we have to use oxygen scavengers, oxygen absorbers in the bottle, high wall thickness bottles can be used. Okay, and then the storage pack can be optimized. We can use the antioxidants and develop the product with barrier film coatings. Blister pack in alu, and that alu blister can be kept into the alu pouch. Select packaging materials based on the oxygen transmission rate and water vapor transmission rate testing and specifications. Then oxygen barrier film coatings for tablet formulations can also be done. Then coming to the photolytic degradation, here I have shown black slide because to have a more impact of this slide. So opaque blisters and bottles with black line inside the bottles can be used. Avoid transparent packaging material. Use colors in the formulation if possible. Then sodium vapor lamps are used in the manufacturing area till the final packaging. Starting from the dispensing to the packaging, we should use sodium vapor lamps. Then specialized coating for tablets and capsule shells for gelatin capsules can be used. Generally, titanium dioxide is kept as an opaquant excipient in the capsule shells and the film coating when there is a problem of photolytic degradation. Also, labeling and storage instruction can be provided that the product required to be protected from light. And also sometimes the instruction is given like keep in the final pack. Then coming to the thermal and metal ions uh, testing and applications in pharmaceutical R&D. if there is a degradation due to thermal and if it is at 40 degree also, then as per the general stability conditions, we study the stability at 30 degree and we give the labeling storage instructions, and sometimes the expiry period is required to be recalculated and confirmed. Then the examples are like store below 30 degree or store at 2 to 8 degree. Then metal ions. If metal ions gives the impurities or presence of metal ions is there, and if impurity get increased, then we can use the metal ion chelators like UDTA is there, and we should use the raw materials which doesn't contain the metal ion impurities, so that these metal ion impurities will not react and will not give rise to increase in the impurities. Then degradation due to humidity and moisture. So if degradation occurs due to humidity and presence of moisture, then generally known fact is the use of desiccants, use of silica gel, molecular sieve, and use of the blisters with very less MVTR. So these are some of the uh, solutions, like select packaging materials based on the OTR and WVTR testing and specifications. Use desiccants and absorbents like molecular sieves and silica gel. Then alu blister or PVC PVDC blisters can be used. PVC PVDC acrylic blisters are also there. Then these blisters can be kept into the alu pouch to have the additional barrier. Also, sometimes the moisture barrier film coatings can be provided to the tablet formulations. Also, use of the solvent based granulation, dry granulations can be used so that the use of water can be avoided. Also, we have to avoid the use of hygroscopic uh, excipients. Some excipients are there which are hygroscopic, so these excipients should be avoided. And the manufacturing should be carried out into the low moisture area or low humidity area. So these are some of the solutions to the problems when there is an increase in the humidity, increase in the degradation of the formulation or API due to the humidity or moisture. So in this video we have covered most of the information about the force degradation studies and its requirement. So whenever deciding the formulation and the manufacturing procedure or manufacturing process for the formulation and the packaging configurations, always we have to consider the force degradation studies and the regular and formal stability studies. It is not like that if any degradation is there in the force degradation due to moisture or light, and it will be reflected into the real time stability studies. So it is not like that, and it is not a general or thumb rule. So always we have to consider both the stability studies. Some of the scientists or some of the people in the pharmaceutical industry, they perform force degradation, they also perform the mild degradation, and also very mild degradation studies, so that there will be a good understanding about the molecule behavior. Also sometimes the force degradation will not give rise to impurity generation, but the mild degradation will give more impurity. So this is strange, but it is a observation for some of the molecules, and hence we have to consider the real-time stability study, the compatibility study with the excipients, and the force degradation studies with harsh conditions or severe conditions or stress conditions and the mild conditions also. And then we have to take a call for the formulation composition, formulation manufacturing process, and the packaging material finalization. So I hope you might have liked the video and its content. Keep watching the videos so that you can improve the understanding, and also by these videos and this platform, my knowledge is also getting enhanced. Thanks for watching the videos. Please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.